Okay, good afternoon, everyone. It's a lovely afternoon here in Nairobi. And I welcome each and every one of you to our today's talk that will be presented by Leo Malingati from our Mammal Committee. I think I should say Happy New Year to everyone. I don't think it's that late because this is a new chapter of this year's virtual talks that we are delivering. So welcome everyone. I just want to let you know that this presentation is being recorded, and after this we will upload it in our YouTube channel so you can revisit when you want to. And also I request that we mute ourselves all throughout the presentation so that we give our speaker ample time. And should you have any questions, should you have any concern or compliments, you can always raise them through the chat box, and we will strive our level best to go through each and every after the presentation. So ladies and gentlemen, you are most welcome. And just to introduce who is our presenter today, our speaker is Leo Malingati Khasoha, and he has a Bachelor of Science degree in Wildlife Management from Egerton University and a Master of Science in Biology of Conservation at University of Nairobi, and currently a PhD student of Ecology at University of Wyoming. And Leo has been a research volunteer at the Mammalogy section of the National Museums of Kenya and a researcher at Mbala Research Center in Laikipia, Kenya. His research interests includes population ecology and community ecology, with special interest in ecology of Kenyan small mammals, that is rodents, elephant shrews, and shrews. And today's title is How Small Mammal Communities Are Maintained in an East African Savanna. Why are widespread species common and restricted species rare? And ladies and gentlemen, it's my honor to welcome our today's speaker, Leo, on board. Welcome, Leo.
Okay, thank you so much, Richie, for the introduction. Just to say, today I'm privileged to give you a talk on small mammals. Small mammals is not something many people study, and we are few who do it. But basically, we try to study small mammals so that we can be able to answer some bigger questions in ecology, or try to move a bit from theories of ecology into realities, like what is really happening within these communities. So let me, okay, so as you all know, life is complex. So is life with every living creature. So life is complex, and wherever there is complexity in life, there is always difficulty in trying to explain what is really happening to things. And so Einstein says, if you can't explain it simply, you don't understand it well enough. So today, if I don't explain it simply, it could just mean I don't understand it well enough. And everyone else doesn't really understand it well enough, because just as I said, life is complex and it is hard to understand it. But I will try to explain it as simple as possible if I can, but bear in mind that there is complexity in life. And so if there is something I don't explain so well, just bear with me. All right, so as I've mentioned, small mammals is not something that many people really do. And one thing you might ask yourself, so what are these small mammals people are talking about? So according to International Biological Program, small mammal is something that weighs maybe from 0.0001 all the way to 5 kg. So if you have a mammal that is 5 kg or less, then that is what we call a small mammal. And there are three categories, major categories that form the small mammals. And number one category is the rodents, which actually makes 40% of all the mammals in the world. So think about any mammal you can think of, like elephants, lions, giraffe, and all that stuff. If you are to combine all the mammals in the world and do percentages like of how many of those are elephants, you will realize that rodents are making up 40% of all the mammals. And then the second group is the insectivores. These are mainly, they feed on insects, though they might eat some other things in their diet, but predominantly they do eat insects. And under that category, we have things like shrews, we have things like moles, hedgehogs, and solenodons. So maybe you might have encountered a shrew in Kenya, a mole in Kenya, a hedgehog in Kenya, but maybe you haven't encountered a solenodon, probably because they are not really found in Kenya. And they are that group of small mammals, they're actually venomous. They have like some venom, just like everything else, like the snakes and so on. So that group produces venom. Like if it bites you, it will let venom into you. And then the last bit of it is the tree shrews, which are not also found here in Kenya. So on the bottom, I'm showing you like a picture of at least one species of each of those groups. So on the left side is a skin shedding mice. We call them, like just know them like as skin shedding small mammals. That is their defense mechanism. They kind of drop their skin, like if you touch them or if a predator tries to touch them or try to pick it up, it will shake off its skin. So just it leaves all of its skin to you, and it can still go and regrow a new skin within a short time. And then in the middle is an elephant shrew. And again, under shrews, we have shrews and elephant shrews. So they are a bit different. And then on the right, we have the tree shrews. So that is just basically the breakdown of what encompasses small mammals. So I hope you can remember we have those three categories: rodents, insectivores, and tree shrews. But in Kenya, we only have like rodents and some species in the class insectivores. So today, I'm not really going to dive into taxonomy and diversity of small mammals that we have in Kenya, but that is something that we can plan and do at some other point. If Richard agrees, or if he feels it's necessary, then at some point we might just take a walk through the wild or even our homes and just see what kind of small mammals do we have around us. So today I'm majorly going to try and answer more of ecological questions rather than doing the diversity of small mammals. So I've been like, if you may ask me maybe what was my interest in small mammals, like I would say as an undergrad at Egerton, I never knew about small mammals being part of wildlife, literally, because when we thought of wildlife, we thought of the mega faunas, like things like elephants, things like giraffes and lions, leopards. But then when I went for an attachment at the National Museums of Kenya, so when you go to mammalogy section of National Museums of Kenya, that is the first place you interact now with small mammals as actually being wildlife. In other words, the megafaunas have overshadowed the smaller mammals, which make up, like, ideally even more than 50% of all mammals in the world. So it is surprising that things that make up just a smaller percent, how they can outshine or overshadow things that make up the bigger percent. So it still tells you there is a lot we can learn from small mammals, and there is a lot that still needs to be done with small mammals. So basically, that's the first place I encountered small mammals. So when you go to that mammalogy section, you will see a lot of shelves full of dead small mammals. We call them stuffed skins. And it will tell you like the diversity of small mammals we have. So that was my first instance. And then I moved to Mpala, where I did a bit of hands-on capture of live small mammals. And I've been here at Mpala Conservancy working on small mammals for maybe roughly about 3 to 4 years now, or 5. So our study, basically we do our study in an experiment that is called Uhuru. Uhuru is not an experiment or a project for our former president Uhuru, but Uhuru is an abbreviation of ungulate herbivory under rainfall uncertainty. So if someone asks you what that Uhuru means, it's not Uhuru Kenyatta, it is this Uhuru. So this is a map of Mpala. Right here I'm showing you like the map of Mpala. And our experiment has been replicated like throughout Mpala, from the northern part of Mpala up here all the way down. And the main idea, or the main goal behind the establishment of the Uhuru experiment, was to try and mimic what would happen in a savanna ecosystem, just in case we have a size-based extinction. So ideally, if you go back to the time of the dinosaurs, you can recognize that we've had like size-based extinction, like things that were so gigantic are the ones that really go extinct, followed by and then they are replaced by things that are a little bit bigger in size than them. So we no longer have the dinosaurs, maybe because they were really big. And right now we have elephant as the biggest animal, and probably it is also facing its own threats. So the idea was to mimic what would happen in a savanna ecosystem if different sizes of mammals went extinct. So this experiment excludes different sizes of mammals. So we have plots that exclude elephants and giraffes, plots that exclude things like dik-diks, plots that exclude every animal, like anything bigger than a small mammal cannot go in. And then we have plots that are just controlled, where everything else goes in and out. And that experiment, when we establish that experiment, when you do an aerial view of those plots, you can see there is a marked difference in vegetation. Like this plot here is an open plot. You can see it is almost open, but this other plot here is a complete exclosure. It keeps off all the large mammalian herbivores, and you can see it has high vegetation cover. And so in my study, I focus on the complete exclosure, this plot and here, and that is where I try to trap small mammals and try to answer a bit of questions that are related to ecology. So enough with our experimental design here at Mpala. I want to move you a little bit into in ecology, there is what we call patterns, there is what we call mechanisms and processes. Most of the time, the focus is always on microecological patterns. Like most studies you find, people are really interested in what patterns can we see in nature. Like, can we see like maybe there is this type of coloration in a certain animal, which is different from another animal that occurs somewhere else. So there has been a bit of concentration on microecological patterns, like the bigger patterns that we see. But then the bit that is always lacking is, can we be able to move from microecological patterns to understand what are the mechanisms and processes that drive the macroecological patterns that we actually see. So assume here on this photo right here, I'm trying to show you like we have two habitants. We have the green one and the white one, and we have different types of butterflies. So from a macroecological pattern, let's assume this is what you see. But then there are lots of questions you can ask yourself from this microecological pattern. Question number one, you can ask, are the different colors of these butterflies, like what explains where the different types of those butterflies occur? So number one, you can ask yourself, are the patterns that we are seeing as a result of abiotic filtering? And by abiotic filtering, I mean, are they like, is it as a result of differences in environmental factors? Is that what is leading to those differences in the microecological patterns that we are seeing? So here, think about the black butterfly, which we can see it is only occurring in the green habitat. And then we have whatever color that is, the brownish one butterfly, it is occurring in both habitats. So you can quickly pick out and say, the butterfly that looks black does not really occur, never ever occurs in a habitat that is white. And then the next question you can ask is, are there biotic filtering? So we've talked about the environmental filtering, and then now we are moving on biotic filtering. And under biotic filtering is where we talk about whether species are co-occurring. And when we talk about co-occurrence of species, if you are a mammal, we are not just looking at another mammal. It could be like, can you really co-occur with a certain plant? Can you really co-occur with a certain animal? Like, how do you co-occur with the rest of life around you? So again, if I take you back to our butterflies, we see our brownish butterfly right here, and this almost yellowish one, they can really co-occur, like they occur at the same place. We can see like this other one here and the black one, they can co-occur. But at no point do we see the black butterfly co-occurring with this less brownish butterfly. That never happens. So you can also pick out a signal about biotic filtering. And then lastly, you can pick out a signal about niche conservatism. And when we talk about niche conservatism, what we mean is, are there phylogenetic correlations in response to filtering? Like, are the differences or are the macroecological patterns we are seeing in the environment just as a result of phylogenetic differences between species, that maybe if a species A, if it occurs in habitat green, maybe it has to have certain phylogenetic characteristics for it to occur there. So basically that is an intro about us thinking from microecological patterns, like we as people maybe who are doing research, or like people who are interested even in life, ideally. I just want you to start to think beyond the patterns you see in the environment, and now try to go a little bit further to think deeply about what could be driving the things that we are seeing. And today I'm glad that I'm going to talk to you about one microecological pattern, and then we are going to try and break it down to understand what could be driving that patterns that we are seeing. So I'm going to talk to you about what we call abundance occupancy relationship. So abundance occupancy just to mean distribution. In other words, what explains the abundance and distribution of mammals or other plants or other, name anything, or insects as we see it today. Maybe you might have gone to a particular place A and you saw like there were lots of animals there. Some of them probably were rare and just occurring in a few places. Maybe some of them were abundant and widespread. And probably you never thought about what could be explaining this kind of a pattern. So in our small mammal studies here at Mpala, we kind of see, we try to check on what is explaining differences in abundance and occurrence of species within Mpala Conservancy. And on the left, I'm showing you a species that is really rare and only occurring in a few places in our study site, and one that is really abundant and widespread, occurring in most of those places. And the question is, what is the difference? Why is the left species not just as abundant as the one that is on the right? And to take you a little bit back again, back into history, it's always good to give a ground to things. So the whole pattern about things being either abundant or widespread, the father of ecology or evolution, Darwin pointed out that dominant species, those which can range widely over the world, are the most diffused in their own country and are the most numerous in individuals. In other words, abundance occupancy relationship is simply one of the most common pattern in ecology. And basically it is the tendency to have things that are geographically widespread to be abundant where they are occurring, and things that are geographically restricted to be rare where they are occurring. And to the best of our knowledge, the first person to point this out was actually Darwin in his book on the origin of species. And ecologists have tried to see whether it is true that when you do a correlation between abundance and occurrence of species, whether you see like there is a positive correlation. So if you check in birds, check in all manner of taxa, or even you try different sampling scales or sampling methods, you will tend to see that things that are abundant, they tend to occur in many different places. Things that are rare, they tend to be restricted in their range of occurrence. And that is not to mean like we might have some species that play different, but mostly the dominant pattern is for those that are abundant to be widespread, those that are rare to be restricted. And taking you to Mpala Conservancy, where I study, I am showing you different mammal species that you can encounter here at Mpala. So when you draw the same thing, like check on the distribution just within Mpala and abundance of these animals, you see that as abundance increases, occupancy of those animals are also increasing, in that things that are mostly abundant, they seem to be also widespread. And there are things that are really down here that are rare at the same time restricted. And this is a macroecological pattern. And the question is, what explains this kind of a pattern? What really explains this kind of a pattern? So these are the small mammals that you can encounter on Mpala. Right here, we have the fringe-tailed gerbil, which is our most abundant species, followed by Hindi's bushrat. And then we have East African pouched mice. And then somewhere in the middle or almost down there, it's the elephant shrew. And then on this other side, we have the Kellen's mouse. It's a dormouse. And then, I mean, this one right here, the dormouse. And then this one up here is the woodland thicket rat. And down here, we have the grass rat. We call it Avicanthus niloticus, Nile grass rat. And down here, we have another gerbil that we call Taterilas harringtoni, which is a Taterilas kind of group of gerbils. So now that we have this pattern within our study area, we need to think about now what are the mechanisms and processes that are explaining abundance and distribution of small mammals on Impala. Number one, there is a hypothesis that was introduced by a guy called Hansky. So he says the differences we see in abundance and distribution, they are basically as a result of differences in rescue effects. What do I mean by rescue effects? So I want you to think about there being a source population, and this source population is providing colonists to the surrounding areas. So we have an area that already has species of, let's say, mammals, and other areas surrounding it that are unoccupied. So what Hansky says, it's the source population that rescues the surrounding areas from extinction. In other words, individuals have to come from the source population and move to the surrounding areas, which sometimes are called sinks, so that to increase the distribution of that species. So if we have a species that is abundant and widespread, an abundant species will provide many colonists or many dispersers, and with that, many dispersers, they will occupy most of those other sites. So we will tend to have many sinks and very few places that are occupied. And on the other hand, if we have a rare restricted species, then it means it has a smaller source population, like it is not really producing enough colonies to move to the surrounding areas and occupy them. So for a rare restricted species, we tend to have many areas that are unoccupied. And Hansky is trying to attribute this to ability of these animals to disperse within the environment. You might start to think or ask yourself, like, why is this really important when we think about it management-wise, or in terms of even wildlife conservation per se? We always talk about there being areas that are important maybe for migration and so on. So we understand sometimes for rare restricted things, migration can just be very important for that species. Like its ability to move through the landscape can just be very important for that species. In other words, like this area that is unoccupied, if you close off migration or close off dispersal, then it means there are no individuals that can come from a source population to this area. And so that means we will have lots of extinctions in many areas for that species, simply because there isn't like dispersal happening. There is no rescue. But if you have a source population and it is connected to many other areas, then we have individuals or other colonists moving to rescue other areas from like those species going completely extinct in those areas. And so we asked ourselves a question like, does the core satellite really explain the shape that we are seeing, like abundance and distribution of species? So one question that we asked was like, we said like abundant widespread species, which I will call core species, these things should have a little bit of low genetic dissimilarity with, low genetic dissimilarity and low population structure compared to satellite species. This was simply to test like, what is the role of dispersal in explaining this relationship? In other words, as you move geographically to the space, like as you move geographically, you should be able to see like things that are more abundant because they are really good dispersers. Their genetic similarity is almost way similar. And then things that are really restricted from dispersing, like you can't really join with another population elsewhere, then we will have these two populations will have totally different genetic diversities. So to do this, what we did was within our study area was to identify 5 species that fall on this continuum of abundant, widespread, rare, restricted. And we were able to collect small distal tail snips, which we used to do a bit of genetic analysis to identify the genetic variability and population structure of these animals. Maybe you might not really understand what's going on the left side, but concentrate on the right side. On the right side, it tells you like we have this species that really showed it had the highest population structure, and we have this one right here that had the lowest population structure. In other words, there was high genetic similarity between populations within this species and low genetic similarity in this species. So when you go back to our relationship here, we see like this animal here, the Jabiliscus, the French-tailed jabil, which is the most abundant and widespread, seems to have the lowest population structure because they are really good dispersers. And this one right here, the Pterylas jabal here, it's down here on this side, and it seems to have the highest population structure. And so that is but evidence for differences in dispersal ability between the rare restricted individuals and widespread abundant individuals. So dispersal by itself, it can bring a total difference. And that's why mostly maybe you hear about people talking about the need for us to create wildlife corridors and so on. It's just because it can really rescue an area that could have gone extinct. It can rescue a certain species from being extirpated in a certain area. And so that is what Hansky envisioned or tried, like this is the mechanism or process that Hansky brought up to explain that relationship. And then there is also another exciting mechanism that can help us understand the differences in abundance and distribution in species. This one, we call it the Brown resource breadth hypothesis. So with the Brown resource breadth hypothesis, all we are focusing on is how are the different individuals using foods in the environment. Like assume if I'm using foods differently from these other species, does that mean we will have differences in our abundance and distribution? So what Brown actually says is that when you are a species that is widespread and abundant, then you must be a species that is using almost all of the resources. So you almost have all the resources you need for you to survive anywhere you can go. But mostly this is a bit of the way mostly we think about how things should be. mostly we usually think like, if you are really good in one thing, then you should be really good in it, and you should be able to outsmart those people who are not able or who do not know how to do that thing that you are good at. In other words, if you are able to utilize one resource and so effectively, then you should be able to attain the highest abundance and highest occurrence where that resource is occurring. But then if you are more of a generalist, like you can eat or you can utilize different things, then you have the disadvantage of not being good at anything. In other words, people call it like, if you are the jack of all trades, then you become a master of none. But if you are a master of one issue, then you thrive in it. So how does this play out in the wild with our small mammals? So what I've just explained is what I'm showing here is, assume you have an environmental gradient and you have a species that can almost occur in all of these places. So this is a species that is more like a generalist. It doesn't really achieve high abundance in those areas. But then if you have another species, let's assume it's the blue one, that is only occurring in a tiny place, then this species attains the highest abundance. I want you to remember the pattern I showed you was abundant things should also be widespread. Rare things should also be restricted. What this pattern will show us, it will show us that things that are abundant, like this is the pattern that this will show us. So it is showing us like actually things that are only occurring in a few places seems to be attaining the highest abundance in the few places that they are occurring. And then things that are occurring in many places, it seems to be attaining just low abundance in those areas that they are occurring. But that is not the pattern that we usually see. The pattern we actually see is that things that are really abundant up here, they also seem to occur way up here. And things that have less abundance are also rare. So what Brown said is like, if you are a generalist species occurring in many places, then you are just able to attain high abundance in all of those places. And if you are only occurring in a tiny place, then even the generalist still outshines you. In other words, the jack of all trades is also seeming to be a master of all. So you occur in many places, and in all of the places you occur, you survive just as better and well compared to things that are even specialized in those environments. So for us to test which one was actually true, whether abundant widespread species seem to show generalism in how they use resources, and whether rare restricted species also show specialism in the way they use resources, what we did was to try and try and understand what these animals are feeding on. So here I'm showing you just a prediction in that if you are abundant occurring up right here, then when we check on your diet, you should be eating most of the things. But if you are rare, then you should just be eating a few of the things. So what we did here was to collect fecal samples from these 5 species and then analyze what plants they were eating and use plant DNA metabarcode to establish that. And actually we have a plant DNA library at Mpala that has about 480 species. So the way this works is that an animal feeds on certain plants in the environment. And those plants, they go through the digestive tract, and finally it defecates, and we can collect the fecal sample of that animal. And that fecal sample from that animal has the plant contents of what that animal fed on. And you can be able to extract the DNA of the plant materials of that fecal sample. And when you sequence it, you can actually compare it to a plant library, in that if you have a plant DNA library, you already know what the DNA of whatever plants you have. And then when you compare it to the fecal sample DNA, you can quickly tell what species this animal has been eating. So again, I want you to remember what Brown hypothesis says is that things that are abundant and widespread, those things should be generalists. They should eat most of the things. And things that are rare and restricted should be specialists. So when you come again back to the findings that we had, we collected all these fecal samples and we tried to analyze and determine what was the dietary niche width of these species. So we realized that things that are abundant and widespread occurring up here, that is again our species, the jarbill, the fringe-tailed jarbill, it had the highest dietary niche width. In other words, it was eating most of the plants. And this one right here, the one that was rare and restricted, it was only but feeding on a few, like it was only feeding on a few plants. Again, when you think about management, think about a rare species that could be endangered. And this species is, let's say, the Taterilla zarringtoni species. And it is only dependent on a few resources, or rather it is only dependent on a few plants or on a few prey. So if something happens to its resource, then it definitely means that species would go extinct. Already it is rare and endangered just because the way it feeds, like it doesn't really have a general kind of a diet, like it doesn't feed on everything. It is a little bit selective. In other words, by it being selective, it is also coming at a disadvantage. So when you think about, let's say, we've heard of stories about, we have just particular habitats that were supporting certain species. And when those habitats like are destroyed, we tend to see like that species just goes extinct. Most likely that species was a specialist. It was only dependent on particular resources in the environment. Once you tamper with those resources, then it's gone. But when you think about things that are a little bit invasive, things that are sometimes problematic, that can attain highest abundance, that can really spread so fast, mostly invasive species are usually generalist. They can thrive anywhere. Throw it on a rock, it will still grow. As long as it can get a little moisture, some little soil, it can still grow. But when you have something that is really like rare, restricted, sometimes those things are really specialized, like in terms of where they occur, what they eat, which somehow just acts as like, which somehow just comes at a disadvantage to them. So we actually see there is support for dispersal, and there is also support for how these individuals kind of use resources within the environment. And so that is the whole bit, like I've tried to move you from the pattern of where we have things that are abundant, widespread, others are rare, restricted, and we are wondering what could be explaining the difference. And we've come down to processes and mechanisms to see that, oh, migration or rather dispersal can explain these differences. Or what they eat and like how you choose foods that you eat out there can really have an impact on on how you attain your abundance and even your distribution. And lastly, I want to quickly take you through one other major pattern that we call the fast to slow life history continuum. And I'm just asking a simple question. This guy here, who is the elephant shrew that weighs just a few grams, are they more like elephants or are they more like rodents? So ideally, when you think about life histories, we have things that are occurring up here, like things like elephants, and we have things that are occurring down here, like the rodents. So most of the time, rodents, they produce many offsprings, they have a shorter lifespan, they grow fast, shorter gestation period, low parental investment, and they have a small body. On the other hand, the other things, they just produce like one offspring, long lifespan, slower growth, longer gestation, greater parental investment, and large body. So we tend to see like there is a continuum, or there is a continuum that is mostly like body size determined. The bigger you are, you tend to be on the other side. The smaller you are, you tend to be on this other side. And actually, when you look at different classes of mammals, you tend to see like things like Proposidae, the elephant, are occurring way up there. Things like rodents are occurring down here. And it is mostly assumed like it's just as a result of differences in their body size. Again, when you correct, but the interesting bit is when you correct for body size effect, you still tend to see there is that positive relation, like elephants are still occurring up here. So the question becomes, if body size is not explaining life histories, then what? So we got a very interesting setup in our study area. We have things that weigh almost the same. So we have the rufous elephant shrew. When you look at its weight, it weighs like about 55. I want you now to compare, like the blue ones here are rodents. This one here is an elephant shrew. When you look at its weight, it's weighing really low. And then its gestation period, it's really just up compared to these other things. Like 60 days gestation period compared to something that has like about 20. Time to weaning, it is just 23 days. The young one starts running about, which is almost similar to the rest of the things. Birth weight, this one has a little bit a bigger birth weight. These ones, the ones have really small birth weight. So, and then this one is producing like 1 or 2 offspring. These other ones about 5 and 3. And then lifespan, it is living 6 years. These other ones are living less than a year, or if they can make it about 1.5 a year. So we are trying to see something that is weighing a really tiny thing that is trying to behave or live as though it was a big animal. Something that is just weighing like a few grams, and sometimes it's weighing lower than other things. And yet 6 years in the wild, just about 1 year, 2 years in the wild. So we asked the question like, what makes the elephant shrew different from the rodents? And basically we went deep to try and check on how their population growth is and what is important to their population growth. So basically, if you had population growth rate, which we call lambda, if you had the population growth of an individual and you had a vital rate, which is survival or other birth rates or recruitment, something that is most important to the growth of that population should be very sensitive to little changes in that vital rate. And then if that thing is not that important to the growth of that, population of that species, then even if that thing varies, there is less sensitivity or there is less change to the population growth. So we have things that live slow, things that live fast. So things that live slow, they tend to show, like when you check the percentage contribution of their adult survival, or rather of survival to population growth, is really high. But then things that live fast, the contribution of their survival to their growth is really low. On the other hand, recruitment for the things that live fast, it has the highest contribution. But for the things that live slow, recruitment is really of no big difference. So in slow living, we tend to see population growth that is really binned around one. One meaning if you have a population growth that is 1 or above 1, that at 1, the population is stable. When it's above 1, the population is really growing. When it is below that, the population is declining. So for slow living things, you tend to see like they tend to buffer what is important to their population growth. So you will tend to see like their adult survival does not really vary with time. And so when you check on what I call coefficient of variation, how variable is like adult survival in slow living things? Adult survival does not really vary, but recruitment can vary because it is not that important to the population growth of these individuals. Like it doesn't contribute a lot. On the other hand, you have things that live fast, the blue ones, and they tend to track environmental variability in resources. When resources are high, or when we have lots of resources, they tend to grow really fast, and then it comes at an advantage. When we have a drought or a catastrophe, then their population just goes down really fast. So they tend to show like boom and bust in their population growth. So remember again, our question was, should elephant shrew be more like elephants, or should they be more like rodents just because of their body size? So this is the kind of prediction I made. I made that adult, for fast living species, and if lambda is most elastic to recruitment, then recruitment should consistently be liable. In other words, it should vary. In other words, for fast living things, recruitment should vary the most. Adult survival shouldn't really vary. For elephant shrew, because recruitment is not that important, it tends to be buffered a little bit. So for this kind of study, we used 3 years of capture-recapture data set, again from PALA, estimated survival and recruitment of these animals, estimated what are the variances in recruitment and survival, and then projected what is the population growth rates per every year for these animals, and then tested to see whether adult survival was being buffered in elephant shrews and whether recruitment could be left to be varying, or in other words, in rodents, like the vital rate that is important to the population growth, how does it behave? And we found that for the things that are fast living, like this rodent up here, and we have the elephant shrew right here that is slow living, when you look at their survival, the survival of elephant shrew is really up at about like 70% of elephant shrews can really survive, but only about 50% or rather up to 45% of rodents can survive. But when you come to like how many individuals it is bringing into the population, you look at the recruitment of the elephant shrew and it is only producing very few individuals into that population, but the rodents, their population recruitment is up. So this one here, it is significant. There is a significant difference in survival between rodents and elephant shrew. There is a significant difference in in recruitment between the elephant shrew and the rodents. And then when you check on quarterly change in population growth, you tend to see like there isn't a significant difference. In other words, we have two sets of animals here. One that is behaving more like an elephant, the elephant shrew, which tends to show less variability in survival, but then it has the lowest recruitment. But still, its population growth is positive. It is still growing. And then we have a rodent, which has the lowest survival, but highest recruitment. In other words, its population growth keeps going up and down, up and down, up and down. But at all the time, the population, when you check on the mean of that population growth, it is still binned to around 1, meaning like the population is still growing. Both for those two populations, like for elephant shrew and rodent, all of them are growing, but they are doing different things. And again, this was just to help you move from a macroecological perspective of seeing things that are maybe bigger or smaller, and how that size-based thing explains things to do in this small mammal community. And so actually we've seen, elephant shrews are more like elephants than they are like rodents. But again, when you check on the phylogeny of them, they tend to share the same glad with the elephants, which is just more evidence to prove, or even what we are seeing genetically, that elephant shrews are way much closer to elephants than they are to the rodents. It is actually true. And so the main purpose as to why I presented to you this talk was to help you see or to help you think beyond the patterns that you just see. Think beyond the things you just see with your eye, to try and ask or question what could be explaining what you are seeing. And we have actually gone through a few mechanisms that can explain what we see from a broader perspective. And lastly, again, the reason I showed you, or I presented to you these, I gave you this presentation, is to show you that, again, as much as the small, tiny animals are so much like neglected or forgotten or seen as less important, but they can actually help us to answer broader questions that have an ecological bearing. Assume if you wanted to study the populations of elephants and follow it from birth to death, probably you will die before you finish your study. But on things like small mammals, you can really answer really fine questions that can help even in management, or rather think of any research. You can actually use these group of animals to answer those questions. So this is simply how we use the small mammals to try and explain bigger questions that have an ecological bearing. And with that, I would like to acknowledge those who work with, that I work with, to collect these kind of data, and my institution, Mpala, National Museums of Kenya, and all these other guys who've been funding our research projects. And with that, thank you, and I welcome any question that you might have.
Thank you very much, Leo, for that very insightful presentation. Please plant elephant shrews and not shrews. Okay, so back to our audience today. The floor is yours. It's time to interact with the speaker. If you have any question, you can, I don't see any question that have come through the chat box, so maybe you can unmute yourself and you can raise the question of the speaker, any comment, any compliment that you have before we bring the session to an end. I see Ivy Ngiru have raised up the hand. Okay, Ivy.
All right, thank you so much, Leo, for a very, very good presentation. It was very, very informative, so that's amazing. I have a question actually, and this maybe we'll have to go back a bit to the hypothesis where you're talking about a population being able to disperse and therefore have better recruitment rates. And my question is, what would be your opinion when you think about some aspects like human influence on rodent populations, and in particular, looking at things like chemical exposure? So maybe, the way we use herbicides or the way we use pesticides, especially in agricultural farms. What would be your opinion? Because I would suspect maybe a population that has very high, it moves out quite highly, is likely to have a strong impact even on wild populations, like the filter population. And this would not be the case for rare species because they are more restricted. So maybe they have less interaction with human beings. So I think I'd like to get that one opinion from you. And then understand also, which is the second one, if we say a rare species has a certain behavior, so maybe looking back into your second hypothesis where we expect a rare species to have a restricted diet or any other aspect maybe we could be testing, do you think such behaviors are fixed, or do you think they're plastic? So, sorry, they have plasticity, I'm sorry. So especially with changes in the environment as we are seeing currently. Thank you.
All right. To answer the first part of your question, one thing that we must appreciate like sometimes rodents within our farmlands, within our residential areas, they are not welcome. We don't like, but when we have those rodents or small animals in the wild areas, then that is where they belong and that's where they should be. Because there are many things like we know rodents can be pests, agricultural pests, and so on. So if someone tries to control rodents within their farmlands, I don't think there is a problem with that. But the bigger problem that we have is the few that we even have in the wild, they are less studied, so less is known about them. And with all things that are, the way things are changing, the way things are even like getting depleted within our ecosystems, then we might be losing things that we even didn't know existed in the first place. Or rather, we have things like small mammals, rather, because they have the fast life history, that these rodents, they have the fast life history, they can really change so rapidly within a short period of time. So whatever you will get later on, that thing has really changed over time severally. So the appeal mostly is just like, as much as we hype for the bigger things, there are also smaller things that also play key role within our ecosystem. The second part about specialism and generalism, yes, what I was simply trying to show you here is that we are seeing evidence that things that are rare and restricted, which are mostly the things that seem to be endangered, they seem to be having quite a narrow range of, like they have low tolerance. They can't just tolerate a huge variation, let's say, in environmental gradients. Like they can't just deal with, let's say, maybe increasing temperatures and so on, meaning those things, they need some sort of urgent attention. So again, here, the small mammals are just trying to show us things that we might never pinpoint. So ideally, like when people talk about things that are endangered and so on, they will tend to stick to just checking on their population, either it is increasing or declining. If it is declining, they come up and say, oh, this species is declining, it is endangered. But when you ask them the question, why is it declining? That is where there is a problem. So you can't really manage a system that you don't fully understand. And that's where, again, things that are fast living can help you to like explore what could be happening even to this other bigger thing.
Thank you. Thank you very much, Leo, for that question. I hope Ivy has been satisfied. I see John Kabue is raising his hand. John, unmute yourself and ask your question.
Okay, thank you, Richie, for the opportunity. Well done, Leo. I think this is really good work. The presentation is very interesting. Actually, I've learned a lot. I thought I knew a lot about small mammals, but I think this was very educative. Now, my question is, of course, I have a lot of interest in the more larger mammals, and I would want to know, maybe from your plots and from your experiments, what is the impact of large mammals, or what is the gradient impact of different large mammals on maybe the abundance or the presence, absence of the various small mammals in the various plots? Maybe where there is presence of elephants and giraffe and so on, are there hind bush rats, are there elephant shrews, or how do they occur, or what is the impact of the large mammals on the occurrence of small mammals? Then on my other question, I would want to know, what is the role of the small mammals in maybe acting as disease reservoir or pest reservoirs, or maybe even disease carriers? Because with the recent incidences of COVID-19 on corona, where we're seeing diseases jumping from maybe small mammals or mammals, bats, and so on. So what role do they play in the whole issue? Yeah. Thank you.
Yeah. Thanks, John, for that question. So about the whole question, I talked briefly about the kind of experiment that we have, where we are excluding different sizes of mammalian herbivores and seeing what impact or what change do they bring on the general savanna ecosystem, so to say. So the plots that keep off all large mammalian herbivores, like things from the size of a dik-dik all the way up, when you compare that with plots that allow everything else to go in, there is a big difference in abundance of those small mammals. So actually you find, like, when you keep off the large mammals from a system, you tend to see like the small mammals, actually they increase in number, like almost triple. They are triple as many compared to the control plots. So which kind of points to something that like the bigger animals could just be helping again to regulate the number of small mammals that we have in our system. Because again, as with regard to your second question, we know like the small animals, let's say the rodents and so on, they can still be quick transmitters of diseases, let's say to humans, especially if they are living way closer to your houses and so on. So if those things can really transmit those diseases that fast, because they are in the wild at the same time they are in your houses. So the instances where man meets with the tiny animals, it's so high. So I think there could be instances where there could be transfer of diseases from small mammals even to humans. But on this other end, when we think of big animals trying to regulate the population of the smaller mammals in the savanna system, one study that was done still here at Impala, it showed that if you keep off large mammalian herbivores from an area, the number of small mammals increased. Like they really increased, like triple the time. And when they increased, you also tend to see like things like ticks, they will increase in those plots. Reason being, the small mammals are just like hosts of these ticks. Like ticks will love those small mammals, and there will be a transfer of ticks from even these small mammals to even livestock. Think of pastoral communities that interact with wildlife. So if you get rid of large mammalian herbivores, small mammals increase. They come with the risk of increased ticks within that system, increased transfer of ticks to livestock, reduced livestock production, which can still affect livestock. And one thing that we usually say is that the big animals, which sometimes people think outcompete their livestock for resources, they could actually help reduce the number of small mammals that would bring diseases even to your livestock. So there is always some sort of a win-win for keeping the large animals in the system, as well as you being able to do with your livestock production. Similarly, another thing, when you keep off the bigger things like elephants and giraffe, you tend to see within those plots, they actually turn from savanna grasslands into some thickets and just bushes, which are not even really important. They are not grassy as such. So, which could also mean that when you keep off larger things, we tend to see things turning bushy, lesser grass, which might not be so good even for your livestock. In other words, the big animals help to engineer the system to keep everything in balance. And so I agree with you, John. Like, we can have issues to do with disease transfer from small mammals all the way to livestock or even to human beings, which is why bigger things coming into the picture regulate smaller animals and keep everything better and good for everyone.
Thank you very much, Leo, for expounding on that. I'm so happy to have John. I remember it's such a coincidence that last year, a period like this, he was also delivering a very powerful talk on larger mammals. Now today, Leo is here bringing us another version of mammals, the small mammals. So I think maybe we'll also create time for two of you in the panel and we do a question and answer session. Thank you very much. Thank you. I really appreciate it. From the chat box, I see there's a question that has come in from Lydia Otieno. And she's asking, for us, we've not understood the difference between buffering and liability, something like that.
Oh, okay. I will help clarify. So when something is buffered, it is kept from huge variation or huge change. When something is labile, it is varying widely. So for fast living things, we tend to see their population growth is quite variable. In other words, it is labile. It keeps changing all the time. But for the elephant shrews, which are more like elephants, their population growth is kind of constrained at one. There is not big variation. And the reason, or the process we said is leading to that, is that for the things like elephants, elephant shrews, they tend to depend most on survival. If they can survive better, their population will increase. It is not all about recruitment for them. To them, survival is the most important thing than recruitment. If you increase the recruitment of an elephant shrew and you don't increase its survival, it will still crash to extinction. So in other words, it's just seeing which vital rate is important for the survival or population increase of a certain species. And why this is important is when you think of management, you need to understand, where should I concentrate the most? Should I concentrate on ensuring that these things are surviving better, or should I concentrate in ensuring that these things are producing more young? So there's no point in you producing more young if recruitment is not significant to your population growth. So that is the whole difference about buffering. So in elephant shrew, population growth is buffered, and also survival is buffered. In fast living things like the rodents, population growth is labile. It keeps changing with time, and also their recruitment keeps changing with time. But the best thing, or the good thing, is that when you check on the average, the population growth of rodents still is at 1, meaning their population can still grow. So it's just a bit of a question of, if there was a lot of rain, will we see something like increasing in abundance? Like if there is, like out here in Impala, right now it is really dry, but when you go out to trap those small mammals, you find like you are finding so many elephant shrews. Reason being, they concentrate on survival. They are surviving better. But right now, when you go out to trap the rodents, they have really declined so badly. You can go out there, you might not find any. But remember, when it rains, they will increase in number so hugely. So they tend to track environmental changes. I hope that helps clarify.
Thank you. Thank you very much. There's a comment from Margaret Vaz. Hi, Margaret. Thank you. Thank you. She's saying, thank you for an excellent presentation. It supports the idea of corridors for survival of the large animals based on your research. Thank you very much for such a comment, Margaret. And I think just some 3 minutes before we bring the session to a close, maybe if there is anyone with a burning last question to the speaker before we wrap it up. Anyone? Okay, thank you. Thank you very much, all of you, for your time and being with us virtually throughout the session. And very much thank you to the speaker for taking us through. So I wish you all a very nice evening, and see you virtually again this particular month, because we are organizing another session for you. Thank you very much, and have a nice time.